diseases and Chlamydiae. In this lecture, we are going to talk about bacteria that are actually obligatory intracellular. So they can grow only inside cells, so they are obligate intracellular. And we have, starting with the Rickettsial diseases. So these are a group of diseases caused by different species. And these are gram-negative Coccobacilli. And what is remarkable about these group of diseases that they share common features, like, for example, transmitting the disease by some vectors. We have ticks, we have mites, fleas or lice. And the ticks, قراد, mites, العث, fleas, براغيث, and lice or louse vector, القمل. So these are the different vectors. And we have what? We have also reservoir hosts. And these reservoir hosts can range from different animals, and also humans can be also reservoirs. Now, in this lecture, we are going to take only those worldwide distribution or in distributed in the Middle East. So in general, these are very small, and they can also be even as small as some viruses. Actually, they were thought to be, some of these microorganisms thought to be viruses. But another thing is also to note that for Rickettsiae, that they can be isolated and can be grown in cell culture. And so there should be some cells in order to grow these microorganisms. And what has been found out that these microorganisms, they can enhance the growth of these microorganisms by applying sulfonamides. And these drugs like trimethoprim, sulfamethoxazole, for example, these drugs can usually enhance the growth of these microorganisms. So this is very important point to remember. And if you see this table here, in this table, we have the disease, and we have the organism causing that disease. We have the transmitting vector, and we have the reservoir host here. So as you can see from this table, we have three major groups. We can classify these diseases into three. So the first one is the typhus. And typhus in Arabic means humma namashiyya, namash, humma namashiyya. And we are going to talk why is it called so. And the other group is the spotted fever, okay? And spotted fever, al-humma al-mubaqqa. And the third one is the Q fever, caused by one single species, the Coxiella burnetii. Now for the typhus group, we have two important species. We have the Rickettsia prawazekiae, okay? And the transmitting vector is the louse, qamil, and the reservoir in humans. And the other one is the murine typhus, which is caused by Rickettsial typhi and transmitted by fleas and balaghith, and the reservoir is the rodents, okay, at Jordan. So this is the first group. Now you have to differentiate. What's very important here is that you should make like a contrast between the typhus and the spotted fever. in terms of the causative agent, reservoirs, transmitting vector, and also clinical picture. In the second group, we have the Mediterranean spotted fever. It is caused by the Rickettsial gonorrhoeae, and it is usually associated with the tick bite. It is the vector here, and the reservoir in the rodents and dogs. While the flea-borne spotted fever, it's caused by Rickettsial felis, and felis, as you know, this is in Roman. It is called the cat. It's also the name for the cats. So the reservoir here, because these are cats and transmitted by fleas. On the other hand, we have the Q fever, which stands alone here as a separate group because it is a bit different. It is airborne fomites. It means the transmissions by airborne or by a vector. And they have more like a larger range of reservoirs, sheep, cattle, and goats. So these are the three major group of diseases. Now we go to discuss each one of them. What is remarkable here, the hallmark in histopathology for the rickettsial diseases in general is the vasculitis and perivascular lymphocytes. And this is usually the cause of the appearance for rash. And these clinical findings of rickettsial infections are characterized by fever, headache, malaise, but most importantly is the skin rash, and usually with hepatosplenomegaly. There will be a history of some vector bite and by the patient, which should be very suggestive. And also the rash appearance can also be very revealing to what kind of group that we have at hand. So for example, in spotted fever, usually the rash appears first on extremities and then it moves toward the center of the body to involve the palms and the soles. It is the other way around in case of the typhus. So this one can give you the difference. And also for the Mediterranean spotted fever, it can be associated with what is called the eschar formation, which is a dead tissue. Okay, so this one can be associated with the eschar. Now, there is an entity of diseases they call Brill-Zinsser disease. And this Brill-Zinsser disease is nothing but a recrudescence of an old typhus infection by Rickettsial prausachii. It is like reactivation of a latent infection. The microbes can persist for many years in the lymph nodes. And just when the immune system of that host, of the human host, is becoming compromised, then there will be reactivation of this infection in the form of a disease called Brill-Zinsser disease. So it's just a reactivation of an old infection. So this is so Brill-Zinsser is only associated with the epidemic typhus, and it is not the epidemic typhus, but it is a reactivation of an old infection. Okay, this is very important to remember. Now, for the Q fever, which is caused by the C. burnetii, the transmissions by inhalation of dust contaminated with the organism. And here in this microbe, we have two different phases. The phase one is the virulent and infectious form of the organism, and it has the LPS expressed, and which plays a key role in virulence. Okay, whereas the phase two of the Coxiella burnetii is not or non-infectious. So the infectious one is only the phase one form. There is also acute presentation and chronic presentation of the disease. Whenever it is more than 6 months, we call it the chronic. If it is below or less than 6 months, then we have the acute disease. Of course, in the blood cultures for bacteria should be negative in the first, usually in the chronic, you don't find the microorganism, but you are going to find the antibodies. And usually these antibodies are raised against the phase 1 form of the microbe. Now, for the Berneti is also the antibodies. They play an important role. Actually, they are the diagnostic method of choice using what is called the indirect immunofluorescence. By the way, the indirect immunofluorescence, it means that we are detecting antibodies rather than here the antigens, okay? There is also some utility for using PCR, useful in diagnosis, and usually in cases where we have chronic phase, where detecting the microbes is very difficult. Therefore, we go for the PCR. This is where the DNA is amplified, nucleic acid is amplified, and we get the result as will be seen at the end because of the amplified product in PCR. Now, of course, diagnosis is based not only on laboratory tests, but clinical findings, epidemiological data, and laboratory tests as well. Now, for the treatment, the doxycycline is the effective, and the one is used and should be started early in the disease. And regarding sulfonamides, they are contraindicated for just the reason mentioned at the beginning of this lecture, because, as we said, the sulfonamides will enhance the growth. So this is a case where antimicrobials, when they are administered without discipline or like sporadically by individuals, this is a good example when antimicrobials will worsen a disease rather than cure a disease. So this will worsen the case and make it just even worse. In case of Q fever, doxycycline is recommended in the acute one, and also other newer microlytes. Microlytes in pharmacology, I don't know if you know or not, these include the erythromycin, azithromycin, and clarithromycin. Okay. And the chronic one is doxycycline and hydroxychloroquine for 18 months is recommended. For prevention, the prevention, as you can see here, because of this disease is dependent on vectors in order to be transmitted. So it is no wonder that controlling those vectors would be an effective method to control the disease by using delousing, insecticides, and killing the rats by poisons. Okay, and in case of spotted fever, we have tick bites can be prevented by wearing protective clothes and using tick repellents. Okay. Now, in case of Q fever, just because it is the reservoir in sheep, cattle, and goats, it makes a lot of sense when you do pasteurization of products, of milk products. And usually short-time pasteurization at 72 degrees for 15 seconds is adequate to destroy viable Coxiella species. Okay, and in case of the Berneti, because it can be communicable or laboratory acquired, there is a vaccine that can be given to health workers to protect them from acquiring disease. Now, we are done with the first one, with the rickettsial diseases. So it's very important to remember this table and how to differentiate between the groups, and also the Brill-Zinsser disease, with which organism it is associated, and the Q fever in case of antigenic structure identification prevention. Now we go to the other microorganism. So this is again an obligate intracellular microbe. And it is also responsible for a group of diseases. It's called the Chlamydiae. So the Chlamydiae are just group of microorganisms with different serovars. When I say serovars, I mean those species that are identified by using certain specific monoclonal antibodies. And it has been found out that some serovars are causing certain diseases. So it's not, so some group of serovars would be responsible for a certain group of diseases in case of a chlamydia. And whenever we talk about chlamydia, you should always remember that it is a major cause for non-specific urethritis. What is special thing about urethritis that's very common in the medical practice, and about 50%, just imagine that, 50% of non-specific urethritis. And when I say non-specific, what do we mean by this term is that non-specific means actually that you don't get any organism isolated in the lab from a urine specimen in case of urethritis. So here, whenever you have a case where the microorganism is not isolated from the urine specimen, then we call it non-specific. There is no specific cause. It's unlike gonococcal urethritis, because in gonococcal urethritis, you're gonna isolate gonococci, okay? So here, the difference is difficult to culture or unculturable. In this case, we call it non-specific urethritis. And about 50% of these cases, which are very, very common in medical practice, are caused by chlamydia. So chlamydia is very important and very common in the medical practice, just to give you an overview and how important it is. Now, you should know something about the developmental cycle that you see now right in front of you. Here we have in figure 1, this image or this diagram. What do you see here? You see the cycle of the chlamydia. They have unique biphasic developmental cycle, and usually characterized by 2 distinct developmental forms. Like we have seen in the C. burneti, where we have phase 1, phase 2, here we have different things. We have, again, 2 distinct developmental forms of this microbe. The first one is called the elementary bodies, the EBs, okay? And the other one is called the reticulate bodies, the RBs. The EBs are all those, the red dots. These are the, here, as you can see here in this part. The cell, and this is the cell nucleus here. You have the cell cytoplasm there, and this is the cell membrane. Now, the first thing to happen, there should be elementary body attaching themselves to the host cell. And this is usually achieved by some molecules, proteoglycans, and also major outer membrane proteins, MOMP for short, and glycosylated MOMPs on the surface of chlamydia. They are also implicated in this interaction between the elementary bodies, those red dots, and the host cell. Now we have the attachment, and the second process is that the second stage, that the EB will enter the cell, because we said before, it is an obligate intracellular, okay, microbe. So there should be entrance inside the cell, and this is usually achieved by receptor-mediated endocytosis. And this will reside in an inclusion. This is the inclusion okay? And these inclusions will usually fuse together, leading to the appearance of what is called the RBs, the reticulate bodies, okay? Now, these initial inclusions will be fused together, as we said, to form the RBs. Now, the RBs, they are different from EBs. This is also very important in the exam, that you should make a difference between them. The EBs are the infectious ones. The RBs are not infectious. This is one very important difference. And also, from the RBs are known to be metabolically active. And therefore, in the metabolically active form, you are going to see more RNA than DNA. Because RNA means there is transcriptions going on. The cell is very busy doing a transcription of the DNA. And as you might be also familiar with the gene expression, that the first thing is that there should be transcription from DNA to RNA. And then the second process or second step in order to have the phenotypes expressed or proteins, that it should be also from these transcripts, there should be translation, and translation is from RNA to proteins. Now, this form, the RB, is very active. It means that it is also has, as let's say, more RNA, 4 times as much RNA as DNA, okay? The EB, actually, it contains equal amounts of RNA and DNA. This should be telling you something. It's not as metabolically active as the RB. And the other thing to happen after that is that, one thing also to remember, that the inclusion themselves, they can give you idea about what kind of species, because we have 3 different species. We have Chlamydia trachomatis, the one responsible for trachoma, and we have Chlamydia pneumoniae, we have Chlamydia psittaci. Okay, so we have 3 different species, medically important. Now, by examining the inclusion bodies using certain stains like Lugol's iodine, because these inclusions, they contain glycogen molecules, so this will be very helpful in diagnosis, because under microscope we can see the inclusion morphology and the color that can tell whether it is trachomatis or not. So if there will be the glycogen, if you see the glycogen by Lugol's iodine stain, then we know that this is a Chlamydia trachomatis by just slide examination, which is very nice, okay? And it's called the glycogen presence in the inclusion. So it is only present in case of trachomatis, while it's not there in the other 2 species, okay? Then after that, there will be conversion because of multiplication of reticular bodies, and there will be enlargement of the inclusion, and then the conversion of RBs to elementary bodies. Here there will be conversion, okay? And after that, there will be release of elementary bodies, and these elementary bodies will go and infect a new host cell. So and the cycle continues. So this is the cycle of development of, let's say, of chlamydia, which is very important to know. Okay, now, as we said, we have different species, actually, and I will just show you the species for Chlamydiae just in a while. And very important that you differentiate between these in the future, or let's say in the exam, okay? And here, and what are the diseases responsible for these species also? So here we are going to see this table just in a while. Okay. So I hope now you see this table. Okay. Now we have this one, the Trachomatis, pneumoniae, and Psittaci. Okay. Now, very important to know that the serovars in the Trachomatis, there are 15 serovars. And because of these large number of serovars, we have different diseases. So we have oculogenital diseases. We have, first of all, the trachoma, right? From its name, Trachomatis. It is the one responsible for trachoma, okay? Which is a chronic keratoconjunctivitis, begins with acute inflammatory changes in conjunctiva and cornea and progresses to scarring and blindness, okay? And we have those serovars responsible for trachoma are the A, B, and C. So these are the ones, the serovar A, B, and C responsible for trachoma. The other serovars from D to K, they are responsible for what is called the oculogenital diseases, okay? And these can affect those sexually active men and women. It is also responsible because of the self-inoculation, because an individual can self-inoculate himself, herself, by getting the microbe from the genital area into the eyes. It is called oculogenital, okay? Because of the self-inoculation, this can lead to inclusion conjunctivitis, it is called. Now, this is an ocular infection that resembles, looks like acute trachoma. And also, when these serovars are acquired by the newborn during the passage through the infected birth canal, it will lead to inclusion conjunctivitis of the newborn. And usually, it is very common, also it is in the medical practice, and it begins with a mucopurulent conjunctivitis after delivery, 7 to 12 days, okay? Now, so from A, C, we have trachoma, D to K, we have ocular, what left? What is left? Now we have the L1 to L3, now this is the L serovars. These are responsible for a disease called lymphogranuloma venereum, okay? LGV here, okay? So the sexually transmitted diseases caused by D to K, we have trachoma by A, B, and C serovars. And also the ocular genitals is also responsible for the infant. But the most important is that the LGV is caused by the L1 to L3 serovars. Now, in case of neonatal pneumonia is caused by usually trachomatis, which is the most common cause actually of neonatal pneumonia. And here, so we have now trachomatis. I think that it's now clear to you how many diseases it is responsible for. So chlamydia is very important. It is also prevalent. And the idea here is that, as I said before, about 50% of non-gonococcal urethritis, they call it non-gonococcal or sometimes NSU or non-specific urethritis, it's the same. It's about 50% of NGU or NSU in men and urethral syndrome in women is attributed to chlamydia. And this produces dysuria, non-purulent discharge, and frequency of urination. Okay, now we go to we have the respiratory. Now we go to another species, which is the Chlamydia pneumoniae. Here, it is only one serovar responsible for community-acquired pneumonia in young adults, in young persons. It's about responsible for 5 to 15% of community-acquired pneumonia in young adults, okay? And it is only one serovar. And it is a similar picture to the pneumonia caused by mycoplasma pneumonia. Now we go to the third species, which is called the Chlamydiae, which is also responsible for a disease called the psittacosis, humma al-babagha. Why is it called so? Because of birds, because of contact with birds, like parrots, babagha, and so therefore its name is associated with that. And psittaci means parrots, okay? So this is usually the contact with birds and airborne bird excreta to humans is the method of transmission. And also here the history of exposure to birds, therefore, is suggestive to have psittacosis. Now we go to a very important topic, is about identification of chlamydia. Given all that disease, all these groups of diseases, now identification is different from according to the type of the disease. So, for example, in trachoma, we have different thing than what we have in ocular genital. And in ocular genitals would be different in case of respiratory infection, pneumonia, and will be also in case of psittacosis, there will be also different methods. So in general, the trachoma usually prevalent in poor areas, as you know. And because these poor areas, they can't afford for the expensive equipment, actually for molecular methods. So it's not the usually it's not applicable in those endemic poor areas. And the molecular methods are the test of choice, on the other hand, to diagnose ocular genital diseases. Why? Why is that? Because it is difficult to isolate the microorganism. It is much easier to go for molecular tests. However, there is very important note here very important to notice that there should be some, given that there will be a proper collection of urine specimen. As you know, in general, whenever you send a patient for a urine specimen to get a general urine exam and culture and susceptibility, you send the patient is instructed usually to collect the midstream urine. Why is it mid? It's mean in the middle. It's not. It means the patient should ignore the first part of the urine, the last part of urine, and should only collect the middle stream. Therefore, it's called the midstream urine. However, this is different here. What you want here actually is the first part of the urine. So there should be a proper specimen collection by collecting the first 20 ml of urine. And the first 20 ml of urine would be fine to have the molecular test, because if you have more urine and you are collecting a larger volume of bladder urine, then this is gonna dilute the specimen, making it even more difficult, and therefore it can lead to false negative results. Okay. There are non-amplified probe assay. Well, this is sometimes it requires some basic knowledge in molecular and immunological assays. Maybe you are familiar with because from immunology and biology, but it's very simple. In molecular techniques, we have either amplified assay and non-amplified assay. In the non-amplified, it means very simple. There is no amplification. There is no increase in the material, initial collected material in the specimen from nucleic acid, be it DNA or RNA. Now, in the non-amplified, we use the immunohybridization technique. FISH, for example, fluorescent immuno or in situ hybridization, FISH, or we use here we use the probes. A probe is a sequence of nucleic acid that is specific to the sequence of the pathogen, okay? And we have the amplified or nucleic acid amplified test, NAAT for short. And these are superior to the non-amplified. Why? Very simple, because now you are amplifying the initial amount of collected nucleic acid, and this will give you better signal to detect, and it can avoid having the false negativity that you just mentioned, okay? The other methods of detection are direct fluorescent antibody, DFA, and enzyme linked immune assay, ELISA or EIA, also by using monoclonal antibodies. Okay. Now, in case of neonatal pneumonia, they can be detected by isolation of the organism from respiratory secretions. Alternatively, serological detection of IgM antibodies against Trachomatis with a titer more than 1 to 1/32 is diagnostic. So this is in case of neonatal pneumonia. In case of lymphogranuloma venereum, the complement fixation is used as a method of detection. Complement fixation, and the rising titer is also diagnostic. In case of Chlamydia pneumoniae, here we have the serology using the microimmunofluorescence. The microimmunofluorescence is a technique using fluorescent microscope to detect the antibody. We can get the titer of either the IgG or the IgM. The IgM for the acute and the IgG for the chronic. And according to the titer or fourfold rise in either IgM or IgG titer, it can be diagnostic. In case of psittacosis, the most sensitive method is serology using the microimmunofluorescence tests. Okay, and also in the lecture you are going to see how to identify confirmed case of psittacosis and how to identify the probable case. يعني أكو case مثبتة أكو case محتملة, probable case. In case of a treatment, usually mass azithromycin treatment is used in endemic areas of trachoma. In case for inclusion conjunctivitis of the newborn, erythromycin tetracycline, if it is not spontaneously resolved. And in case of the genital infections by Chlamydiae, we have sexually transmitted diseases. The general principle is that you don't treat the patient that presented to the clinic, but you also treat the partner. It's very important, and sometimes the offspring. And also you look for other diseases that can be associated with Chlamydia, like usually it is associated with a group of diseases that are transmitted or communicated at the same time. So we have bacterial, viral, so you have different microorganisms that can be transmitted at the same time. This is also a possibility that should be kept in mind. And also, in case of the neonatal pneumonia, oral erythromycin can be given. In case of LGV, lymphogranuloma venereum, can be achieved by sulfonamides. Okay. The Chlamydia pneumoniae is sensitive to macrolides in general, tetracyclines and fluoroquinolones. Okay. And in case of psittaci, the treatment is based on the clinical findings. I mean, without the need for laboratory tests because of the difficulty to get laboratory confirmation. And treatment is with azithromycin, clarithromycin, erythromycin. These are called also the macrolides. For epidemiology and control, we have trachoma has a long history of getting more people infected and with blinded by this infection, about 400 million people worldwide. So it has a very large burden. The WHO initiated called the SAFE program, SAFE. This is very important. The S stands for surgery. Why? Because we have scarring and deformity of the eyelids. So surgery for deformed eyelids only. And the A stands for azithromycin therapy, and the F for SAFE is the F, face washing and hygiene, and also the E is the environmental improvement, like building latrines and reducing flies that feed on conjunctival exudates, like control of the flies. Okay. And also we have the preventive measures in different other diseases. And like, for example, psittacosis, because the birds are the ones, also the reservoirs, therefore controlling birds and should be very important, and screening them for diseases, control of bird shipment, quarantine, testing of imported birds for psittacosis infection, and also they can be given prophylactically tetracycline as a control to this disease. So this is the end of the lecture, and I mentioned the main point. Thank you for listening. And now we go to questions and answers if you have any questions. So thank you. So do you have any questions about the lecture? Otherwise, we are going to have discussion meeting at 8 o'clock today. Also, I would like to remind you of the quiz tomorrow. Or on Saturday. Wait a minute, it is scheduled. So okay, so you have on Saturday at 4 p.m. we are going to have the quiz on 3 first lectures, okay? The microbiota and the gram-negative, okay? And so the first 3 lectures, okay? So we are going to have also quiz on these lectures. So do you have any question about this lecture? Okay. You're welcome. Okay, thank you for coming, and please fill out the attendance form. Okay.
Okay, Doctor.
Okay. So until next time, thank you.
دكتور
Yes.
بالنسبة للدسكشن يعني نقدر نغير موعد الدسكشن يعني إذا أكو إمكانية.